These are the all-important products of sunbeams, the green leaves. And they're important because you can eat them. At least some of them. This is spinach. And not only can you eat them, you must eat them to stay alive, to get the energy to stay alive. Either them or the things which are derived from them, like the fruit and the sugar and the starch and all the other goodies that one gets out of one's food, like the cake. Would you like to join me in a piece of cake? How about you? Now I hope you're going to... Go on. I hope you're going to enjoy it, because I warn you, you're going to have to do a little work to earn it. Do you feel the energy coming into your stomach? Do you know how you're getting that energy? Well, the starch and the sugar is reacting with the air and the oxygen and creating the energy. Let me show you how it does it. If I take a piece of this cake, it isn't too bad, is it? And then I dip it, give it plenty of oxygen by dipping it into liquid oxygen. And then I hand it to Mr. Coates. He'll show you how cake reacts with oxygen. Well, it reacts quite well, doesn't it? Do you feel that happening in your stomach? Because that's just what's happening in your stomach. The cake and the oxygen are giving out energy like that. How's it feel? Well, there is a slight difference. That reaction went at a temperature of something over 1,000 degrees, and that's very bad for the digestion. So the reaction that went on in your tummy went on at a lower temperature. And that's because nature has provided us with catalysts to make these reactions go. A catalyst, lots of catalysts in chemistry. A catalyst is any substance which makes a reaction go better without itself taking part in that reaction. For example, here is some hydrogen peroxide, which we make in our stomach when we're digesting foods. And although it's good for some purposes, such as if you want to be a smashing blonde, it's not very good in the stomach. In fact, it's a poison. And so in the body, and in this is the remains of my liver and bacon breakfast, in liver in particular, there is a catalyst called catalase, which when put into hydrogen peroxide, decomposes it very quickly. Hydrogen peroxide is H2O2. And this catalyst breaks it down into water and oxygen. Now, let me tell you something about, that's a biological catalyst. Let me begin by showing you a catalyst with which you may be more familiar, a catalyst which is used quite a lot in industry is platinum. Now, you know platinum is a very valuable metal, and we can't afford lumps of it at the Royal Institution. Besides, people tend to take them home as souvenirs. So this is platinum on the surface of asbestos, so that we have a lot of surface. And if this is not lit, this is a gas flame, and if I put this into the gas flame, you see it glowing. It's mainly hydrogen, this gas, and you see how it's glowing red hot in the flame, although the flame wasn't lit, and eventually it will light the flame. And that's because it breaks down the hydrogen into atoms. It's not changed itself, and enables them to react with oxygen more readily. Now let's return to the biological catalysts, and they are called enzymes. Now, I showed you an enzyme last time. Let me first of all just show you sort of schematically how an enzyme works. Suppose this is something like hydrogen peroxide. This is a molecule of hydrogen peroxide that we want to break down. And it doesn't break down very readily. And if I sit it on any old surface, it doesn't break down either. But if I sit it just on a very specific shape of surface, then it breaks down very readily. And the same is true if, this is always true of catalysts, they catalyze the reaction both ways. If I want to put these together again, they don't go together again very easily. But if they're sitting in the right position and I put them together, then that enables them to recombine. Well, that's how a catalyst works. And it's a sort of lock and key mechanism. You see, it's an exact fit between the substrate and the catalyst. And now let me show you a real one. And this is one which you saw last time, one we're very fond of, lysozyme, because it's the first enzyme catalyst whose structure was ever determined, and it was determined here by David Phillips. Now, this is typical of an enzyme, and the thing about it, it was discovered, incidentally, by Fleming, Sir Alexander Fleming, who worked in London, and he got a cold, which is not uncommon for somebody who works in London, and a bit of mucus from the end of his nose dropped onto a plate he was preparing, a culture, and he found that where the drops landed, all the bugs had been killed. And it was lysozyme that was doing this. It also occurs in tears. In fact, one method of manufacturing it is to beat somebody hard until he cries, and then you collect the tears. But you can also get it from egg white and other sources. Now, you see, this is, I don't know whether you remember, it's made up of a chain of 129 amino acids, which we have hanging up there, and they fit together in this exact structure, and there is a hole in the structure. Do you see that? There's a gap in the structure there. And that gap is the key to open this lock, which is the sort of sugar molecule which makes up the cell wall. And that's what has to be broken down by this. And that fits absolutely precisely in here. It makes a lot of hydrogen bonds in there, fits absolutely precisely. And when it's there, snap goes one of those bonds. And that's how these catalysts and these enzymes work, and that's how we manage to break down the cake. And the equation then, let me write an equation. The equation we've got to so far, chemical equation, is that the chemical substance called cake reacts with oxygen and gives us energy. And if we'll try to establish all these facts, and we'll see how much energy these two have in a moment. And it presumably gives us something else as well, because you can't destroy matter, and we're going to see what that is now. Now, is your cake nicely digested? would you like to come out and help me to try to find out what else we get from this cake apart from energy? Now you're Louise and Richard. Can you just stand in front of it so that I can get at these? Now, these are, we are going to do two separate tests for carbon dioxide, because I have reason to suppose that I'm going to test anyway whether the product is carbon dioxide. This is lime dissolved in water, calcium oxide, and you see it's just like water, it's colorless. And this is an indicator which changes color when it goes orange, when it becomes acid. So now, of course, we've got to burn this cake of yours yet, haven't we? So will you run right up to the top of the theatre and down again as quickly as you can and burn the cake? Off you go. Right up to the top. And then I'm going to get them to breathe into these. I should think that's burnt a tiny bit of the cake. You've a long way to go yet. Now, will you take that one, and you take that one, Louise. You come here, Richard. Now blow away the products of the reaction between cake and air, cake and oxygen. And I had two controls, exactly the same as the ones we started with. And when they've finished blowing, we'll see whether there's any change. You can breathe in as well, in between, if you like. Come on, puff, puff, puff. Breathe in. Well, Louise's is gone. Let's have a look at yours first, Louise. Would you like to... Oh, you stand where you were then, and you just show us those. You see the difference? Now, that's gone milky, and she has... This is... Can you guess what it is? Carbon dioxide and lime has made... It's just chalk, calcium carbonate. So that's one proof that you have combusted the cake to carbon dioxide. And how's this one going? All right, Richard, let's compare those two. Hold them up. Put them against your nice white. Now, I think you'll see that's gone from, what would you say the one on the right is?
Orange.
Orange, and the one on the red is still it's red. Yes. So that's two proofs that you've made carbon dioxide. Thank you very much. You can go and rest and digest the rest of your cake. So I think we can safely put carbon dioxide there. Now, cake, the main part of it anyway, which is going to give us energy, is carbohydrate. You remember we had a lot of sugars and starches and things, and they have the formula 1 carbon and 1 water. That's why they're called carbohydrate. So we have carbohydrate plus oxygen, let's write that out, plus oxygen gives energy, plus CO2, and you would like to balance your equation. What do you think is left? Water, thank you. And if you add up everything, you'll see that is the complete equation. And that's what happens when we eat the leaves, or the products of the leaves. And now, the question is, where do all these goodies come from? Because this reaction supplies all our energy, not just our food, but our fuels. Every bit of energy on the earth is supplied that we use, with minor exceptions like nuclear energy. All that energy is supplied in this way. And the answer is, of course, it's supplied by this reaction being driven backwards, carbon dioxide and water from the air make us let us call this starch, for example, make us our starch and oxygen. But energy has to go in too, and where does the energy come from? Sunbeams. Let us just put light. And perhaps I should write that backwards for you now, because this is what we're really going to be talking about. Carbon dioxide and water from the atmosphere under the influence of sunbeams giving us CH2O. You realize, of course, it's CH2O many times, many C's, but it's in that proportion of 1 carbon to 1 water and oxygen. Now, I like to try to prove everything to you. We can't prove everything, but we'll try to prove as much as possible. Let us now try to show that we do get oxygen and we do get starch when we carry out this great process which is called photosynthesis. Here are some green pondweed, and I'm shining light on it, and I'm going to try to show you that oxygen bubbles off. You've probably seen in your pond, if you look at pondweed, that there are bubbles. Sometimes the bubbles bring it up to the surface, and that is oxygen which is coming up to the surface. And it's a slow reaction, but I think if you look here, you will see bubbles coming up to the surface there. And if I tap it, you'll see them come up a bit faster. You see all those bubbles coming up there? This has been running through the morning, and you can see a gas is being formed. Of course, I haven't definitely shown you that is oxygen. So let me now do this by the good old traditional. It's not going to bang. It's not oxygen and hydrogen. It's just oxygen. The traditional chemist's way of putting a glowing splint into it, and you see the glowing splint gave a little flash as it went in. It didn't leap into flame, but it's still glowing inside there fairly well. So that we have now established that we have oxygen, and we want to establish that we have starch. And Elizabeth here is going to help me with this experiment. Elizabeth left school only a few months ago, didn't you, Liz?
Yes.
And she's here helping us with these experiments before she goes to Oxford. Now, what we're going to do is to take a leaf, take a geranium leaf, for example, and we're going to put it into acetone, right?
Ethanol.
And this is ethanol, into
Yes.
ethanol, like that, and leave it for about 10 minutes, which is too long. So she's going to show us one which has been treated in that way. And you see it's lost most of its pigment here, and it's become almost colorless. Now, this leaf that she's taken has been, the geranium plant has been looked after very well, and it's had lots of sunlight, so it should have made starch. And now we're going to test for starch. So Elizabeth has put it in the dish, and she's pouring iodine onto it, because iodine and starch form a nice, very dark blue compound. And whilst that's going, let me show you that. Here is a reaction which makes iodine, and there's a little starch in it. Takes about, I made it so that it takes about 10 seconds to make iodine, so you'll be able to see it clearly. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's the color, and this is developing. You see a dark color, and it will eventually go black. Now, I want to show you a little trick you can do, because you might like to do it yourselves. You can have a lot of fun this way. If you took a geranium plant and covered part of the leaf up for a long time, starch would be formed where the light was falling on it, but the starch wouldn't be formed where it's dark, would it? So if you do that and then put it in iodine, only the part that was in the light will go black. You see what I mean? Now, Professor Walker in Sheffield does some beautiful sort of science art tricks in this way. He takes a leaf, and this is what I suggest you might like to do. He takes a leaf, puts some glass in front, and a black card behind, and a bit of blotting paper. Soak the blotting paper in bicarbonate of soda, because the plant likes carbon dioxide, as you know. And you have a little hole so that the plant is happy and can be put in water. You see, like, thank you, Liz, as we're doing here, we've put the stem in water. You can, in fact, do this with a living plant without taking it from the leaf, without taking it from the plant. Now, either by putting a negative over it or by using your little home projector, you can project a picture onto it like this, you see. And only where the light falls will starch be formed. And you leave this, how long do you leave this for, Elizabeth?
3/4 of an hour.
About 3/4 of an hour. And this is the result. This is one that we did a couple of days ago. And you see, there's the picture we printed. It says Royal Institution, and there is a sun smiling down on all of us. Well, thank you, Elizabeth. You might like to try that and try some, see what sort of pictures you can make. And if any of you feel that you have produced a masterpiece, bring it along and we will show it to everybody else in a later lecture. So now, we have this wonderful, wonderful cyclic process of nature. This wonderful recycling whereby the energy, nothing happens except the sunlight provides energy for Richard and Louise to run up and down stairs. Nothing else happens. The carbon dioxide is recycled and the starch is eaten, and it's all a lovely cyclic process. For example, animals and plants can live together. Here's a sort of representation of our world, a representation of our world of animals and plants. Here, these little mice are in a totally enclosed jar, and they're living with plants, and providing there is plenty of light falling on the plants will produce the oxygen for the mice will produce the carbon dioxide for the plants, and everybody will be happy, like us, who do the same thing with plants. Some of you may have done this at home, put plants into a bottle and stoppered them, completely stoppered them. And there again, they live quite happily, because in the dark, plants are like animals. They can also breathe in oxygen and burn food and produce carbon dioxide. In the light, they do the opposite process, and they strike up a nice balance so that they can live. And Bill Coates here planted this garden. It's looking a bit tatty now, Bill, I'm afraid, but he planted this 5 years ago. And the stopper, I assure you, is fixed, because you can come and try afterwards to get it out. It's stuck, in fact. We can't get it out. That's why we've had it for 5 years. Otherwise, we might have planted a better garden. All right. Well, now our next question must be, what is it that absorbs the light, and what does it do? This is a wonderful chemical manufacturing machine, the leaf. And we would dearly like to know how it works, because it's interesting, but also because we would like to do the same ourselves, perhaps even do better. So the first thing we must ask is, what are the green things? What are the pigments in the leaf? And again, Elizabeth's been helping us with this, and I'll show you how you can find out what these pigments are. We're going to use grass, are we?
Yes.
The best botanists use spinach, but we're going to use grass because it's more readily available. And that you're pouring in is acetone. And you see, this is really just a bit of cookery. We have a perfectly conventional blender, and we stir it. We chop it up, in fact, for about 20 seconds or half a minute. It's quite enough. And that gets out more than half of the chlorophyll. And then Elizabeth will filter it, leaving all the rubbish behind. And it'll go through. Okay, that's all right. A nice clear green liquid coming through here, and then she will analyze that and separate it. Let me just show you how that is done. You remember last time when we were talking about amino acids, I mentioned chromatography as a method of separating the amino acids. Well, let me show you a little more show you how chromatography is done. This is a plate of glass which has been coated with alumina, and I'm going to dot onto it here a mixture of inks. Let me get my ink. They're a mixture of red and blue inks, and I'm going to dot those onto here. I could just draw a pen across if it worked a bit better, but if we dot it like this, this will do equally well. All right. Now that's a mixture of red and blue inks, and I'm now going to put it into that solvent like that, which happens to be ethyl acetate. Take that away, Bill. I always knock it over. Okay. And now the solvent just rises up by capillary action, rises up the alumina, and I think you can see already, perhaps, that the red is beginning to rise ahead of the blue. And we'll come back and have another look at that in a moment, but certainly the red is separating from the blue, and that's the principle. If you have a mixture of substances, they are swept along at different rates because of the different attraction they have for the alumina. Now, you can do this in a number of ways, and a common way is to have a column of alumina, the same stuff, aluminium oxide, and pour the mixture on the top, and then pour the solvent on and let it sweep down. And Elizabeth has done this here. This was set up this morning, wasn't it? And it was just a matter of taking some of this solution here and putting it on the top. And you see the separation that you get here. Here is one chlorophyll. Here is another chlorophyll. And here is a nice yellow band. It's rather pale, but if you can see it, that's a nice yellow band. That is carotene. Do we have carotene? We don't have carotene, do we? Does anyone have carotene? What have we got? No carotene. Is there just one molecule there? There's carotene. Here we are. There we are. Thank you. Stand up with it. We've got some molecules tucked away so we can identify them. That yellow is carotene. It's a double single bond. That's why it's colored, and that's carotene. Thank you. And the other ones are chlorophyll, and it's chlorophyll that is all important. And that's how we separate. We can chop out this column now, you see, and get the pure chlorophyll. And when we do, we find this molecule. This is chlorophyll. This is what caused the Green Revolution. If you like, this is the cause. This is the molecule that matters. The other molecules, carotene, for example, is in carrots. You wouldn't have guessed that, would you? Carotene is in carrots. Xanthophylls, which come out as well, are in the autumn leaves, the yellow and brown autumn leaves. When the chlorophyll disappears, that's what's left behind. But in all of these plants, what matters in all of these plants is chlorophyll. It's the chlorophyll which is the basic molecule which does it. Chlorophyll is a fairly complicated molecule, I suppose, but the only thing I want you to notice is that it has an oily tail, a paraffin tail, then it has this ring, and it's this ring, porphyrin ring as it's called, which does the absorbing of light and makes it green, because it absorbs what colors? Everything except green. Thank you. And it has also in it an atom of magnesium, rather unusual, but quite a lot of biological molecules like to have just one atom of a metal in them. Now that chlorophyll is common to all of these things, from the simplest green algae, this is chlorella, to which, incidentally, you remember the most primitive green plants first appeared on Earth 2.5 billion years ago. That was the beginning when the oxygen first appeared. And incidentally, let me tell you that although I described how the oxygen appeared by the photochemistry in the atmosphere, that was only the beginning. Now, that reaction I've been talking about is the one that's mainly responsible for making oxygen. And in fact, all the oxygen in the atmosphere, every molecule of oxygen in the atmosphere goes through green leaves every 2,000 years. It's all recycled every 2,000 years. And even all the oxygen in the sea goes through the green leaves every 2 million years, which is a long time, but not long in evolutionary, in comparison with the age of the Earth. And so from the simplest algae to remember the plants, the plants of, the land plants only appeared 300 million years ago, relatively recently. All of these things, all shapes and sizes. This is the taro plant. The Hawaiians like eating the root of that. It's called poi. And it tastes like awful gruel, I think, but all of these things are green leaves. They all work through chlorophyll. Now let us ask a little bit about the structure. Let's look at one of those leaves through a microscope. First of all, let me show you a very simple alga, such as, in fact, this is Euglena, which we saw under the microscope last time. And it's sort of half plant and half animal. It swims around, and yet it's self-sufficient because it synthesizes its own food. And here we have the little round blobs which contain the chlorophyll. Now you can see those better if I show you a green leaf. This is a bit of a green leaf under a microscope, and this is one leaf cell. And if you look in that cell, can you see those little round things there? That's where all the action is. Those are called chloroplasts. And in fact, you can tear this cell to bits so that you just have the chloroplasts, and it will still do the job. It will still do the job of photosynthesis. Now let's take one of those chloroplasts. Well, first of all, let's take a cell from which we've taken the cell wall and look again. Here is another picture of a cell, and you can see the chloroplasts in this one, rather big, and sticking to the cell wall. And what they look like is, here's a model of a chloroplast, like the one we've just seen in the picture. These black blobs are the grana, where most of the chlorophyll is. And if I look round the other side, I see the product that we're all after. Those are blobs of starch which have been formed by this chloroplast. So, all right, well, let's blow this up a bit further now. Let's have a look at one of these chloroplasts here under more magnification. This is a stereo scan picture taken with an electron microscope, and you can see the chloroplasts rather nicely there. Let's blow them up further. Now, this is a chloroplast, and you can begin to see those grana that I talked about, that you saw in the model, and you can see some lamellae, can't you? You can see some lines which contain the chlorophyll. Let's blow that up further still and take the membrane away so that you can see better. And now we're really beginning to see something about the structure. We're down almost to the molecular level now. We're down to the molecular level, and you see these lamellae, as they're called. Very, very thin indeed. In fact, they're double, and each one of the double parts is only 1 molecule thick. And I'd like to show you how they're arranged. Remember, they consist of chlorophyll molecules to some extent, but of lipids. Now, who's the fatty boy? Who's the fat? You are. You don't, perhaps. Could you stand up, hold the red bit? No, the red bit, and you write, hold it up, that's right. Now, show us the 2 tails. Can you show us the 2 tails? Sort it out, Bill, will you? It's got a bit tangled up. This is lecithin. It's a lipid called lecithin. Just hold. That's right. Hold it up by the. Now you see, it's got two tails, and it's got all these red parts, which are oxygen and nitrogen, and blue for nitrogen at the top. That's what lecithin looks like. That's what fats look like. Thank you. And I want you to now reduce your scale a little bit, and that's what he was holding up. No, we're always jumping scale so that we can see different levels of information. Now I want to show you how those are fitted. At least the idea at the moment, the best idea on how those things are fitted together, these lipids, fats, chlorophyll and so forth, are fitted together not only in the chloroplast and the chlorophyll membrane, but in cells and in our bodies, is as follows. Molecules like that put their heads together and their tails together like this and form a layer, one molecule thick. And then the oil dissolves in oil, doesn't it? And water doesn't dissolve in water, and oil doesn't dissolve in water. So if you have two layers, the oily parts get together. And that's why you get a layer 2 molecules thick. And then the protein, the enzymes, all the bits of the molecule where the action is, are gripped into that. And no doubt, we don't know by any means all the detail about this, but they're gripped in like that within this molecule, and it's in that sort of structure that this wonderful chemistry takes place. Now we must begin to look at this wonderful chemistry and ask, what happens after a photon of light comes along and hits the leaf? What's the first thing that happens? We have chlorophyll. Here's some of the chlorophyll that Elizabeth extracted for us from the leaves. And you see it's green, very dark green, and that absorbs the blue and the red light. Let's suppose it absorbs the red light, because red light works. What happens next? Well, the next thing that happens is that if nothing else happens, it just gives out the light again. It fluoresces. And I've got two other substances here which fluoresce, so we look at all of them. Let's shine some dark blue light on them, in fact, ultraviolet light on these. Take the light, all right, that's okay. And you see they all fluoresce rather nicely. And I hope you can see that the chlorophyll fluoresces a beautiful red. Although it's green, it fluoresces a beautiful red. You can see that here quite easily in the theatre. You don't see it on the television screens because the cameras, at least these cameras, are not responsive to red light of 680 nanometers, which is as far as this goes. Now the question I want to ask is, how quickly does that excited molecule stay? Now I'm going to remove the light, and I want you to tell me how long it takes for the fluorescence to disappear. Up, Bill. Not very long. All right, lights, please. Okay. Not very long. How long would you say? Well, it's 5 billionths of a second. Did anyone count that? No. So we have to use rather special methods to do so. Let me just show you what's happening, first of all. Let me just show you what's happening. Remember, we saw this model before. But now what happens is, you must now imagine this was an atom. This is now a chlorophyll molecule with its electron here in an orbital. A photon of blue light comes along and is absorbed, and the electron goes up. This is the excited electron. Now it sits there looking around for 5 billionths of a second, and then it emits a red photon. It does that, thank you, it does that because whilst it's up there, it loses a little bit of its energy as vibration and to the solvent and so forth. Well, what we would like to do is follow the course of that excited molecule. That excited chlorophyll up there with its electron excited is somehow going to produce the energy in the leaf, which we're going to eat. And we must follow it through this course. But if it's going to do that, it must do it in less than 5 billionths of a second, mustn't it? Because otherwise it'll lose the light. And this is, I want now to spend a few minutes telling you how we can follow that excited molecule through not only a few billionths of a second, but a thousandth of a billionth of a second. We do this with lasers, and downstairs we have a laser which enables us to do measurements in a millionth of a second. We call that a picosecond. I want to take some chlorella from, yes, in a little cell, thank you. And Colin Treadwell here is our specialist who works down in a laboratory right in the basement, which is directly opposite Michael Faraday's laboratory, which is still there. And I think even Michael Faraday would have been rather surprised to see what he does sometimes. Well, I'm going to give him this little specimen of the chlorella, and he's going to take it down to the basement, because I can't take all of you down to show you that, because the whole room has to be air-conditioned. It isn't much bigger than this space. But Colin will take that down whilst I explain what goes on. He's very kindly made for us this model of a laser. Let me just show you what a laser is. Thank you. A laser, the kind of laser we're using, consists of a rod. This is ruby. That's just one piece of ruby made synthetically. And that ruby rod has chromium atoms which are excited by a flash of light. This is a flash lamp. The ruby rod goes inside the flash lamp, and when the flash goes off, the ions are excited, and in fact you excite more than you leave behind. So you get an inversion of the population, as it's called, of the excited molecules, and they then begin to excite each other. They stimulate the emission of others. You get a chain reaction, rather like an explosion, and out of the end of that rod comes a pulse of light. And so this is the little laser there. Since you can't see our very special laser downstairs, we brought up an old-fashioned laser here, which has George Snovitz, who's operating it, looks terribly hurt when I say that. But the thing is, this one produces a pulse of only a billionth of a second, a few billionths of a second. But nevertheless, it'll do. And just to show you that it does produce a pulse, he's going to fire. You'll notice we have some balloons, a blue, a red, a blue, and a red. And he's going to fire it now. And it does what it should do. That's... What color do you think that flash was? Quite right. Because, of course, the red balloons didn't absorb it, did they? And it's a red flash from a ruby laser, and it lasted, the pulses in that last about 20 billionths of a second. Thank you, George. Now we'll go back to... Now, let me show you what happens in our picosecond laser, which is mode lock. Colin has made this apparatus to show what happens. Here is the laser, and we have a mirror on either side of this laser, and we have a dye here which bleaches every time it holds off the lasing action, and then when a pulse hits it, it bleaches, so that you get an oscillation of the light backwards and forwards between these mirrors. But then this mirror isn't a perfect mirror, so some gets through. You see, like this. It goes through a frequency. The light that comes from the laser we use downstairs is in the infrared, so we have to make it to a lower useful frequency, and we do this by doubling its frequency and making it green. That's why you can do this. If you have very high intensities, you can stick two photons together and get double the frequency. That's called frequency doubling. And so when it goes through the frequency doubler, it's green. You see the red changes to green there. And then it hits the chloroplasts, the chlorophyll, the leaf that we've just taken down there. And when it hits those, they fluoresce red, as I showed you on the model, and go into the streak camera. Now, the streak camera is not a camera for photographing streakers, as you might think by its name, but it is a camera for streaking across a very fast message, so we can see how fast it is. You see that little light there? Looks continuous, doesn't it? But if I move it quickly, if I streak it across your vision, then can you see that it isn't continuous? Can you see that it consists of pulses? Those pulses are 1/100th of a second each, and you can probably see the length of the pulses. Can you? So if we took just one of those pulses and streaked them through the streak camera, we get a message which tells us how fast it is. Now we're going to do that. Colin took that down, he has put it into the apparatus, and we're going to do the whole experiment here with a bit of luck. We've got a link, and Colin, you there?
I'm here, Prof.
Good. Well, now I wonder whether one of you, Richard, would you like to? All right, you come and do this. Now here's the button, which is going to fire. Colin will do nothing. He's got it all set up. Could you don't press the button yet. Put your thing, and would you hold your hand there so that we can all see you press it, and press it when I say. Now, what's your name?
Christopher.
Now, when Christopher presses that button, he will fire the laser onto the chlorophyll. You won't see very much, but I hope you will see on this screen the fastest experiment you've ever seen, and certainly the fastest that Christopher has ever done, because we shall see the excitation rise and decay. Have you charged up, Colin?
Yes, the capacitor's charged up.
All right, good. Would you like to count Christopher down so he knows when to press?
Certainly. 5, 4, 3, 2, 1, fire.
Good. And there it is. Did you see that jump up? On the front side here is when the pulse fired. It excited the chlorophyll, and the chlorophyll now decays like this. And one division on that tube is 60 picoseconds, 60 millionths of a millionth of a second. A picosecond is so short that light only travels 1/3 of a millimeter. And the Concorde would just about travel across one molecule in a picosecond. So it's a very, very fast process indeed. Thank you very much, Colin. Just to show you perhaps a little bit better, when we have a little more time, we get rather better pictures. Here's a picture of one pulse. You see it's 9 picoseconds across there. We can select one pulse from that. And this is what you saw on the screen, only rather better done. This is chlorella. And you see it starts at 50 picoseconds, and it's half down again after another 7. It's only 70 picoseconds for this to decay. Now that means that when the light strikes the leaf, it excites the chlorophyll molecule, and in 70 millionths of a second, it's finished. Not the whole process, but that particular process. Now, I could tell you a great deal now, I would love to, about what follows. I'm only going to tell you very briefly about 2 things, and I think perhaps not all of you may understand quite what I'm going to say, because I'm going to have to abbreviate it and say it rather rapidly. But I'd like to give you some idea of the excitement of what is happening in this field at the moment, because it's a terribly interesting and important process. The first thing I'd like to tell you about is I'm going to tell you 2 very clever things, you see, that nature has invented in the leaf. And the first is what's called light harvesting. Nature has invented a process whereby it doesn't matter where the energy is absorbed, it doesn't matter which chlorophyll molecule absorbs it, it moves around until it lands just where the leaf wants it to be, like this. The photon, here's our photon, is absorbed by any old chlorophyll molecule like that. These are some lipids which got in. It's all right. And that would excite that chlorophyll molecule so. That's excited. You can see it's excited. Now, that excitation, the photon is gone, but that excitation can wander around extremely rapidly. In the time of that experiment, in a few picoseconds, it can wander around, and it will continue to do so until it lands on this special chlorophyll molecule, which has a slightly lower energy, and there it's trapped. And that's where the action is going to be. That's where the action is going to start. Now this wandering around from one molecule to another, also from the carotenes to the chlorophyll, from one pigment to another, that occurs by a resonance. I've used that word before. Let me show you better in this context what I mean. Suppose these are two chlorophyll molecules and neither of them is excited at the moment. They are pendulums and they're the same frequency. Now if I set one pendulum swinging, that's just like this one now begins to swing, you see? And it's transferring its energy from that to that. And eventually it'll be complete. And now all the energy is in this pendulum and this one has stopped. And now it goes back again. And this one will transfer its energy back to this one. And so the energy oscillates backwards and forwards. That's called a resonance transfer. And if you think of those as chlorophyll atoms with the electrons going around, and you think of having a lot of them, rows and rows and rows of them, you can see how the energy is transferred in this process. Now what happens here? We're now at the trap, and now we're ready for some chemistry. Well, what happens is electron transfer. Here I have a blue dye instead of a green one, chlorophyll, but I have something which will donate an electron. Chemistry in biology is very much electron transfer from one molecule to another. And if I shine light on one of these, the other one is just for comparison, there's no deception here. If I shine light on this one, you will see that very quickly it's bleached, and that is because the molecule has transferred its electron to the dye. But then it comes back again. You see how it's coming back? What's happening is as follows. Here is my blue dye, and here is my iron with an electron it's happy to give up. If I excite the blue dye by exciting that electron, then it can pull over the electron from the iron, like that. But now that will drop down, it'll be a normal molecule, and if they now come together again, the electron will go back. That's what happens in an electron transfer reaction and the back reaction. Well, now, in photosynthesis, we have to transfer 4 electrons. We have electrons and hydrogen atoms chemically are almost the same thing. We have to transfer 4 hydrogen atoms. I have 2 water and a carbon dioxide. To bring about the reaction I want, I have to transfer 2 hydrogen atoms to make the water and 2 more hydrogen atoms to make the carbohydrate, and that gives me my products. 4 electrons must be transferred. And very quickly, I want to show you the basic scheme of how we believe that is done in photosynthesis. This is called the Z scheme, and it's the sort of map on which all people, and there are hundreds in the world working on photosynthesis, all people discuss what is going on. Our problem is as follows. We have to transfer 4, this is energy up here, all right? This is energy, and we have to transfer upwards in energy 4 electrons from water to carbon dioxide. That's the difference in energy between them. Now, if this is a red photon, and the height of that represents its energy, you see it isn't big enough. And water doesn't absorb red light. So chlorophyll does, the leaf does it in the following way. There are 2, this light, remember when I showed you how the energy is collected? That's called a light harvesting unit. But there are 2 kinds of light harvesting unit, which we call kind 1, type 1, and type 2, 2 units. Now, this is type 2, and that has a chlorophyll molecule, remember our trapped chlorophyll molecule in it, and this is type 1, and that has another chlorophyll molecule. And what happens is the following now. these can absorb a photon of light, and the electron can be excited. And this one can as well, and the electron can be excited. And now, this can drop down to there, that can drop down to there, that can drop down to carbon dioxide, and what's the net result? We're back where we started, except that we have transferred one electron to carbon dioxide. 2 more photons, another electron by the same process. 2 more, another, 2 more, another. And that is how it's done very cleverly. No chemist can do that, so that eventually, of course, we finish with oxygen and our carbohydrate. No chemist can do that. The leaf has discovered how to do it. We would dearly like to be able to use 2 photons in series as the leaf does. Well, now, that's about as much as I can tell you about the details of this process. There are many, many intermediates in that scheme. But to finish with, I'd like just to ask one question that perhaps you've been asking, some of you. And that is, I said this was a forwards and a backwards reaction. One was reverse of the other, but it isn't, is it, really? Because if I burn a fuel in oxygen, I don't get out light, I get out heat. I don't get light, sunlight, sunbeams. Well, not normally, and nature doesn't do this much, but it does it. One little beast that has managed to learn to do it is the firefly. Of course, the female firefly. Naturally, it's the female that's the flashy one. And the female sits in the tree and flashes to attract the boys. And she does this by chemistry. She does it by carrying out a chemical reaction, which is exothermic, which gives out energy, and excites a molecule, knocks an electron up to a higher state so that we can get cold light. So these are not the substances the firefly uses, but they're not dissimilar. They're organic substances which carry out a reaction. And if I mix these 2 substances, you'll see what the firefly does. As those substances are mixed, we get chemiluminescence, cold light, which is formed by the chemical reaction exciting an electron of a dye stuff, exciting an electron of a dye stuff, and then the electron drops like this. Well, there aren't many uses for this at the moment, although there might be. But when you came in, some of you were given a little tube like this, which we're asked to put under the seat. If you'd like to take it out now and look at it, this is a mixture of 2 substances rather similar to the 2 that I've just had. It isn't a mixture, it's 2 substances which have not been mixed. And when they are mixed, they will give out light like this. And it is of some use for map reading. It's useful if you want, it's very safe. If you want to go down and see where the gas leak is, it's the safest way you can use. So if you would all break them, just like that, and shake, you will see the chemiluminescence of the same kind as we saw before. It will light you on your way home. It won't last all night. But my foes and my friends, it gives a lovely light.